Thanks everyone for joining. So in today's 3D Genomics 101 presentation, I'll walk you through what is 3D genomics, what role does it play, how is it studied, and how is the data visualized. Then I'll walk you through some case studies and finish off with how you can access 3D genomics through Dovetail assays and a little bit about the Dovetail difference. So the fundamental question is, how does a 2-meter long DNA fit itself into microscopic cells? And the answer to that is 3D genomics. And 3D genomics is that deliberate packaging of the genome in the nucleus. It's based on the fact that the genome is not randomly organized and is not randomly packaged in the nucleus. It actually has a very deliberate organization, and it's an organization that shows hierarchy. So at a high level, you see those chromosome territories. And each of those territories is the region that each of the chromosome occupies in the genome. And you can see that in the nucleus. And you can see that those territories or those regions are not overlapping. So the chromosome territories is the non-overlapping regions that each chromosome occupies in the nucleus. If you zoom in a little bit more, you see that the chromosome is organized in A and B compartments. A are those active chromatin that is actually showing a high level of transcriptional activity, versus B that is gene-depleted, repressed, and closed chromatin. If you zoom in more, you will see TADs, or topologically associating domains. And these domains promote interactions within regions that are contained within the domain, and limit interactions with the regions outside of that domain. And they play a role in gene regulation because, to put it more plainly, if you have an enhancer, having this TAD domain or this TAD organization will limit the ability of that enhancer to activate and interact with a promoter that is located in a different TAD or a different domain. And if you zoom in more, you're going to see the loops. And the loops is that physical folding that is bringing in regulatory elements, such as an enhancer, together with a promoter to activate the gene expression and recruits the transcription machinery. So this is a different level of the organization that you see. And now to address, why does that organization matter and what role does it play? 3D genomics play an important and active role in cell biology. It plays a role in gene expression, because it's going to make it either easier or more difficult for the transcription machinery to access the DNA. So it will influence the gene expression and the activating and the repression of the gene based on this accessibility of the transcription machinery to the DNA site. It also plays a role in chromosome stability, and it helps maintain the chromosome structure during cell division. So you're having less likelihood of a breakage or a genetic material loss when you're having that cell division. So it maintains the chromosome structure. It also helps the cell respond to a DNA damage. So it makes it more easy for those repair proteins to access the damaged site and repair it. It also plays a role in epigenetic regulation. So this chromatin remodeling, which is this dynamic change in the chromatin folding, is one of the epigenetic regulation mechanism. It's going to make it, again, easier or more difficult for the transcription machinery to access a DNA site. And this is how it's going to regulate gene expression. This is more from an active standpoint of how 3D genomics, or what role does it play in cell biology. But there's also a lot of leverage that genome assembly researchers take from 3D genomics data, because it helps them understand what parts of the genome are located together. So what sequences you can locate together, they come from that same chromosome. How are those regions oriented? How are they ordered? So they're using the 3D genomics data to assemble the different contigs or different parts of the genome in a chromosome scale assembly. And then they will use that assembly as a platform for a lot of downstream analyses that help feed into their evolution, conservation strategies, developmental biology. So it has a lot of downstream benefit. So how is the 3D genomics studied? There are different techniques for studying 3D genomics, one of which is one of the most prominent technique, is chromatin capture conformation. or 3C technique. And this techniques, there are also different flavors of it, but all the 3C techniques, they have in common this core workflow, which uses proximity ligation. And the idea behind them is that you're actually capturing interactions between regions of the genome that are co-localized in the 3D space. So they're interacting with each other in the 3D space, but that they are distant one another in that linear physical space. So that's really what proximity ligation is. It's allowing you to ligate and interrogate the interactions that are happening in the 3D space between different regions of the genome. And the core workflow is this one here. It's a genome-wide approach. So with this core workflow, what you're asking is actually, let me catalog all the interactions that are happening across the entire genome. So this is how you get a genome-wide 3D data. And the way to get to that data, you will first start with your sample. It can be cells, tissues, or blood. You will cross-link your sample in order to preserve the 3D structure of your genome in the nucleus. And then you're going to fragment that cross-linked chromatin, and then you will perform the proximity ligation, which you're actually going to ligate fragments into these chimeric DNA molecules. So what you're having here is a chimeric DNA molecule that is linking two regions of the genome that were co-localized in the 3D space, but they don't come from next to each other in that physical linear space. Now that you've generated proximity ligated DNA molecules, you're going to convert them into sequenceable libraries that you can sequence, get your 3D data, and run that cataloging of the interactions that are happening in your genome. This is the genome-wide approach. The other flavors are more targeted approaches. So they're built on that core workflow, but it depends on what you're targeting. So you have this first flavor is when you're targeting interactions that are anchored with your protein of interest. So really here, what you're saying is you don't really care about all the interactions happening in the genome. What you care about is what are the interactions that are happening with your protein binding sites. And for that, what you will do is after cross-linking and fragmenting your chromatin, you're going to carry chromatin immunoprecipitation. So you're going to precipitate on your protein of interest, and then you're going to perform proximity ligation. And what you're going to convert into a library are these interactions that were happening with your protein binding site, which are associated with your protein of interest. So this is one flavor of the targeted approach. The other flavor is more a sequence-centric viewpoint. So unlike the protein-centric viewpoint, here you're really focused on the interactions that are associated with your regions of interest. Usually that region of interest is a promoter site, but it doesn't have to be. It's really any region of interest that you're looking at. And the way you do that is by first generating this library, this whole genome library, and then you use that as input for hybridization. And you will hybridize with probes against your regions of interest. And that's the library that you're going to sequence, which referred to as a capture library or an enriched library. So you're really now looking at the interactions associated with your regions of interest. Moving on to how is the data visualized? So in the intro, I was sharing with you a little bit about these organization of the 3D genome, which you can also refer to them as topological features. So AB compartments, data loops. But this slide focuses a little bit on, what you would expect to see once you have your 3D data and you're starting with the analysis. So the AB compartments, again, as a reminder, A are the active open chromatin, B is that repressed closed chromatin. And when you see on that contact map AB compartments, you're seeing this checkerboard pattern. And the reason for this is domains from the same compartment preferentially interact with each other. But you don't have the A and B domains contiguously located on the genome. So what you're seeing is an alternation of A and B domains. To put it in other terms, what you're seeing here is that this domain and this domain don't really interact much with each other. So this domain and this domain don't belong to the same compartment. In contrast, this domain and this domain interact more with each other. And the way you're seeing the interactions is with the intensity of the color. That's how you're really seeing a contact map. The contact map allows you to visualize the interactions that are happening, and you're capturing that with the different color intensity. And the more frequent those interactions are, the higher that intensity of the color that you're going to see. So these two domains don't really interact with each other that much. So these two belong to different compartments, while this domain and this domain interact with each other more frequently. And that means that this domain and this domain belong to the same compartment. It's the same thing you're seeing in the Genome browser, which is another way for you to visualize your 3D data. So what you're seeing here is that the blue color belongs to the same domain, the red color belongs to the same domain. So you're having these two compartments. One is in blue, one is in red, and you have this alternation of the A and B compartments across your genome. So they're not located next to each other in the genome. That's how you're seeing this checkerboard pattern. And that's how you see on the Genome browser this switch between the different compartments. TADs are usually visualized as a triangle. So what you're seeing here is this is one TAD, this is another TAD. And as I mentioned, by definition, TAD means that you're having regions in the same, contained in the same TAD, interact more with each other. That's why you're seeing this intense red color. Then they interact with regions outside of the TAD. So here you have few interactions or less frequent interactions between these two TADs, but you have a lot of interactions that are contained in the same TAD. And if you're seeing this in a Genome browser, what you're seeing is these rectangles. So what you're seeing is this is a TAD, this is another TAD, this is a third TAD. And it really, the TADs act as barriers. So to put it into context, if you have an enhancer here, having this TAD is going to limit the ability of the enhancer here interacting and acting on a promoter that's located in a different TAD. And this plays a role in gene regulation. Loops is this apex that it's usually seen as this apex of the triangle. And what this is saying is that this region and this region of the genome are interacting with each other. So there's that physical folding that's bringing together this region and this region. When you see it on a Genome browser, you're seeing the same thing, just a different representation. So what you're seeing is these arcs. And what that arc is describing is that, again, this region and this region are interacting with each other. And you're seeing multiple arcs depicting or cataloging all the interactions that are happening between the different regions in the genome. And usually at that level, the interaction is really between a regulatory element, such as an enhancer, with a promoter site. So a little bit on, real life examples of 3D genomics and how it helped identify or address biological questions. So a little bit on the format of these case studies. Each slide will represent one research project. It will highlight where that work was published. So you have that journal link here, the date it was published, and it will start with a little bit of a high level for what is the research question that lab is working on. Then it will go into what specific question they're trying to address and how 3D genomics help them address that question. And we're going to differentiate between whether they use the whole genome topology or whether they used actually a more of a targeted topology, whether it's protein-centric or sequence-centric targeted approach. So starting with Bart Decker, he set on to understand what is the function of the mini chromosome maintenance complexes in DNA replication, which, a very important process for the cell. And what he found out using whole genome topology, so cataloging the interactions that are happening across the entire genome, he found that those mini chromosome maintenance complexes act as obstacles that impede the formation of loops. So what you see here is the wild type, and you see this loop exclusion that is facilitated by the cohesin complexes, which is here depicted in these yellow circles. And this loop extrusion, when it sees the MCM complexes that you see here in a red hexagon, it's actually blocked. So upon encountering these MCM complexes, this loop extrusion is blocked. This is what you see in a wild type. in the samples that had the MCM knocked out, so you actually don't have these MCM present, you see that this barrier is actually lifted. And that's why you see that loop extrusion that can happen. So, with that, the conclusion, or what 3D genomics data was able to show, is you're actually seeing a lot more loops formed when you don't have the MCM complex present compared to the number of loops that you're seeing in wild type, where MCM is present and it does act as a barrier to that loop extrusion that's facilitated by the cohesin subunit. Another case helps in understanding and identifying structural variants. So structural variants is this rearrangement of large fragments in the genome. And 3D genomics actually enable researchers to identify a structural variant that's happening in their experimental condition. So here, that's a contact map that we were seeing as a cartoon earlier. And Sun Rhee from University of Southern California used the whole genome topology approach to study the different structural variation that's are happening in prostate cancer. And you can see here with this butterfly pattern, they were able to identify from this contact map that actually there has been an inversion that has happened in these cancer cells around this gene, ARID1A gene. And this gene is known to be dysregulated in prostate cancer cells. So not only they were able to identify that there is a structural variant, and the structural variant is an inversion, they were also able to identify the formation of novel loops. So what you're seeing here in this blue circle is loops that were formed as a result of that structural variation. And those loops, what does that mean is they're actually going to bring a different folding of the chromatin. They're now going to bring an enhancer next to that promoter site, and they're going to regulate the expression of the genes that are involved in the prostate cancer. So this was enabled with the whole genome topology. Another case, this is more using a targeted approach. So now we're talking about protein-centric topology by Arul Shanayan and Abhijit Parolia from the University of Michigan. And what they were set to study is the role that SWI/SNF chromatin remodeling complex plays in the dysregulation or in gene dysregulation in prostate cancer. So it's also in the oncology world, and we're looking to understand this complex specifically. What role does it play, or how it's actually influencing the dysregulation in prostate cancer? So it's known that it plays a role, but here it's more about understanding the actual mechanism for this dysregulation. And what they were able to identify is that this SWI/SNF complex alters chromatin accessibility, and it allows the chromatin or the transcription machinery to access the DNA site, the protein, the promoter site, and activates the expression of genes that are involved in the cell proliferation and maintaining and sustaining the tumor cells. So this SWI/SNF complex, when it's present, it allows the transcription machinery to access these oncogenes or these DNA that code for proteins that are involved in the overexpression or proliferation of cancer cells. Then they treated these samples with PROTAC. It's drug-mediated treatment. And this drug specifically targets the SWI/SNF complex. When the SWI/SNF complex is impaired, this transcription machinery is then not recruited to these sites. You're losing this activation of these oncogenes. And as such, you're downregulating the expressions of genes that are important in the proliferation and the maintenance of cancer. So this shows a promising therapy strategy for prostate cancer. The last case that we're showing is more related to genome assembly. And this focuses on how this whole genome topology information was used to help this researcher, Maria Vittoria Carminati from the University of Colorado Boulder. They helped her build a reference genome. So that is a chromosome scale genome that is important for her study on subspeciation and citation cancer development in humpback population. So having a reference genome, a high quality genome, is important for her to be able to study these processes. The current short or long-range read sequencing did not enable her to achieve that chromosome level or chromosome scale assembly, but using that in addition to this whole genome topology enabled her to actually assemble the contigs in a chromosome scale, help inform the orientation and the ordering of these sequences, and end up with a reference genome sequence that she's able to use for understanding a little bit more about the subspeciation and the cancer development in this population. So as I mentioned, for all of these cases, we have the link to the journal if you wanted to read more, but we wanted to just provide a little bit more of like biological questions that our researchers and users of this technology are asking and how they're going about addressing them. Now, how do you access the 3D genomics? You're able to access them through either using a kit that we supply or through our services. So we have different options depending on your research question and the application that you have in mind. And this table just outlines a little bit of the high-level description of the different assays that we have. And as I mentioned, these assays are available to you as kits or services. So TopolInk is an assay that we've recently launched, and it allows you to have a whole genome high-resolution topology very rapidly. So you're able to actually complete the workflow in a single day and have the data that allows you to understand the 3D organization and the chromatin topology of your sample. MicroC is more used to understand the chromatin topology at a nucleosome level. So this is really when you're trying to address the question of nucleosome positioning or nucleosome interactions. Our MicroC assay is best positioned to do that. Our high-chip assay and pan promoter, these are assays that are more targeted approaches. So the high-chip speaks to that protein anchor topology. It's going to allow you to generate a library that has the interactions associated with your protein of interest. The pan promoter is more of a sequence-centric approach, so it will allow you to generate data that has interactions associated with your region of interest, and in this case, it's actually your promoter regions. OmniC and AssemblyLink, these are assays specifically for genome assembly applications, and they support genome scaffolding, pseudohaploid scaffolding, haplotype resolved scaffolding, that is diploid scaffolding, or even polyploid scaffolding. So, whatever you're looking at for your genome, OmniC and AssemblyLink will allow you to get that chromosome level assembly. And AssemblyLink is a rapid genome scaffolding assay. So you're also able to get your data and analyze that, to get that data for analysis in a single day. And what we pride ourselves on is that Dovetail Difference is the fact that you're actually able to do that 3D genomic study in the most accurate, less biased or unbiased way that's possible on the market. So the traditional way to generate 3D genomics data has been based on a restriction enzyme. So it's known as restriction enzyme-based Hi-C methods. And the bias that incorporates is the fact that you're really only digesting or resolving your genome around those restriction enzyme sites. So you're going to underrepresent sequences in your data if these sequences come from regions that don't have a lot of restriction enzyme sites, and you're going to overrepresent other regions. So what you're seeing here in red is that traditional Hi-C, and you're seeing that you're having underrepresentations of regions where you're blind to that sequence because you don't have a restriction enzyme site, or what we call restriction enzyme deserts. Whereas when you're using the Dovetail assay, it uses a sequence-independent approach for that digestion. It actually uses enzymes such as DNase, TN5, or MNase that don't rely on a restriction enzyme site. And what you end up with is this coverage that's uniform, unbiased. You're covering your entire genome, and it's very similar to what you get from a shotgun library. And that has different implications or various implications. One of them is you're actually able to generate or capture biology accurately. So when you're calling your topological features and understanding the chromatin organization, you're not distorting that based on the location of your restriction enzyme sites, which is what you're seeing here. You're actually capturing that topological feature at that biological site because you're not biased to where a restriction enzyme site is present. Also, because of this very high signal to noise ratio that these assays offer, it helps reduce your experimental, your cost, either through sequencing or what it takes to prep the libraries by more than 12-fold per data point. So you're actually going to save on the cost for sequencing and the cost for library preparation because of that high signal to noise ratio. The other bit I wanted to cover is that the different informatics solutions that you have. So it's not only providing the ability to generate the library, but also, helping you with what to do with the library once it's generated. You have different options. You have the self-serve options. So these are documentations, workflows that you can actually run at your end, or we also offer services where we run pipelines for you, or you can consult with one of our bioinformaticians to help customize the informatics or the data analysis to what your research needs are. So it really scales and it speaks to the needs of your lab. I think with that, I'll leave you with where to find more information. You can speak with one of our experts, download our ebook that talks a lot more about these different techniques for studying 3D genomics and why 3D genomics is important. You can learn about our products, either the kits that we offer or the services that we offer. And we also have a really interesting webinar about 3D genomics and why it matters that you can also just view as a self-serve. So we have the links and we have these codes for you to find out more information.